We have two seminars each week. Welcome to the Immune seminar. I'm super excited to introduce Shan Liang as our speaker today. Shan Liang obtained his bachelor degree from Nankai University, and then he went to Johns Hopkins University, study with Robert Silicano, a really giant in virology and HIV infections, who discovered the latency of HIV infections in human. Through Shan Liang's PhD, he really became an expert in antiviral response against HIV. Then he took his expertise to Yale in the laboratory of Richard Flavall, continuing his research endeavor of studying HIV infections and antiviral response. In addition, in Richard's lab, Liang expanded his interest and pioneer works in humanized mice. In 2017, Liang started as assistant professor in Washington University in St. Louis, Department of Medicine, and continuing his long-term interest in virology and immunology. Throughout Liang's career, he has been exceptionally visionary and productive. While he was a postdoc, he authored several significant publications as the corresponding authors. And just a couple months earlier this year, he published two landmark works on Science, identifying cellular sensors for HIV infection, which we'll hear today, and also pushing the humanized mice into a new era. So Liang, welcome to the seminar, and we look forward to your talk.
Hi, Xu. Thanks for the nice introduction. Everybody hear me okay?
Great.
So my title is a little bit specific to a specific inflammasome sensor, but I want to talk about general, interest in the intersection of HIV and immune response, because this is still a puzzle that will continue, I don't know for how many more decades. Let me move on to this slide, which really is a very telling slide. So these are the two pandemics that everybody know. As far as SARS-CoV-2, within less than a year and a half, due to its superior transmission capacity, there has been 176 million people infected. But also owing to our own immune system and perhaps vaccine, 159 million people already recovered, but still 3.8 million lives have been lost. But then from this figure, we know that our immune system can dominate the battle against SARS-CoV-2. By contrast, HIV is much more lethal, or has a much higher mortality rate. This year marks the 40 years since the AIDS outbreak. And in the past 40 years, we estimate that there have been 75 to 80 million people infected by HIV, and nearly half of them already died. And we could have lost more lives without antiretroviral therapy, because more than 99% of the infected individuals will potentially progress to AIDS and die without treatment. And so far, there are only 2 patients cured of HIV infection, and both of them received bone marrow transplantation, a procedure that removes all the white blood cells because these patients develop leukemia. So this is not a generalizable approach to treat or cure HIV infection. Therefore, there are 38 million people currently living with HIV, and they will be living with HIV for their lives if there is no cure strategy developed. And hopefully for the SARS-CoV-2 pandemic, in a year or two, it will be hopefully completely behind us, and there will be absolutely no people with lifelong active SARS-CoV-2 infection, very different from HIV. So then I think the major difference is the viral infection and our immune clearance. Like for RNA viruses like SARS-CoV-2 or influenza viruses, the acute infection will drive robust immune response and clear viral infection normally within a week or two, and the outcome is clearance. And then with the understanding of the natural immune response, scientists and industrial partners can design vaccines to mimic the exposure of the viral antigens to trigger immune response. And in people receive vaccine, what you expect is the pre-existing immunity can quickly clear viral infection before any symptom develops. However, if there is no functional natural immune response, it makes the vaccine design really difficult. And another example is HBV or herpes viruses. They can establish latent infection so that they cannot be fully cleared by our immune system, but they are largely under control by our immune system. The last category is HIV. Obviously, we know that our immune system are not equipped to win the battle against HIV. So despite very robust anti-HIV specific immune response, you don't see the viral clearance or you don't see the viral suppression. And then the persistent infection eventually drive to AIDS progression. So people always believe that, retroviruses and RNA viruses have very high mutation rates. So that's the issue posed against our immune response and also posed against vaccine design. So at least by in vitro culture system, if you compare all the microbial pathogens in terms of their mutation rate, the bacteria, because the DNA replication is more faithful, so the mutation rate is much, much lower than the viruses, especially the RNA or retroviruses here. And HIV and influenza has very similar size of genome and also very similar rate of mutation, at least in vitro. But then for influenza, the very different part of the infection is actually these viruses rarely mutate in humans. So the virus normally undergo a lot of antigenic shifts or drifts in the wild reservoirs. However, a selection of the pandemic strain in each flu season normally is determined by how the virus can escape the pre-existing immunity. But once the pandemic strain is established, they don't really mutate much after they enter the human population, and which is also true for SARS-CoV-2. So then if you see this phylogenetic tree collected in isolation year of 1996, worldwide strains containing 96 sequences of the HA, and you see this is the size of the tree, the area indicates the total mutation. So by contrast, if you forget about the in vitro culture, you sequence HIV from patient, then what you get, the DNA sequence from HIV, you can compare with the in vitro culture mutation rate, you can see a 2 to 3 log increase of the mutation rate, suggesting that HIV mutation is accelerated in patient, which is also true if you look at this phylogenetic tree. So this is again the 96 worldwide HA sequence from the influenza. And then if you, one scientist collect HIV sequences in the 1997, this is the 9 HIV envelope sequence from a single patient. And the area of the tree is as big as the sequence collected worldwide for the entire flu season. And then if you collected 193 of these HIV sequences from different patients, then you can see that the HIV is much more diverse. And this diversity is mainly driven because the high intrapatient mutation rate. As you can see here, there is not such selection of pandemic strain. Any given HIV strain has the full capacity to establish infection and rapidly mutate and evolve in any single human host. That drives really the overall viral evolution. And that's why our immune system or the vaccine makes it very difficult to overcome such a highly diverse viral population, no matter within a single patient or within the infected populations or high-risk populations. So what really happens is that during primary infection, the virus trigger a robust immune response, which partially suppress viral replication until the escape variants quickly emerge. and then the drive the shifts in the dominant immune response. And these cycles go on and on, leading to persistent infection. And we know that the persistent HIV infection will cause the systemic loss of our CD4 positive T cells, which is the hallmark of AIDS progression. So we often ask the question, how can we help our immune system to win a battle against HIV? And so far, we still don't have the answer. That's why for any HIV-infected individuals, or vast majority of the HIV-infected individuals, they have to take antiretroviral therapy. And the drug is really, effective. That's really suppressed the viral replication within the first couple of months. And then the viral load in the plasma can be quickly below detection limit. However, ART or ART does not cure HIV infection because the virus can hide in resting CD4 T cells as a piece of integrated genomic DNA. And then these CD4 T cells harboring integrated proviral HIV virus, the frequency is about 1 to 16 cells per million total CD4 T cells. So for each ART treated patient, they carry average 1 million such cells carrying replication competent virus. And because these are resting memory CD4 T cells with relatively long half-life, average 44 months. So if you allow the viral reservoirs to automatically decay, that takes about 70 years, suggesting that the patients on ART have to be on ART for life, because these reservoirs can sporadically produce viruses. And because the presence of ART, they cannot rekindle infection. But as soon as the patient stops ART, the virus rebound within normally a few weeks. So that's why ART cannot cure HIV infection. It can treat, but the patient have to be on ART for life. And our work, together with other groups' work, showing that although the ART can reduce the frequency of HIV to a very low level, but still our immune system cannot control the viral rebound, suggesting that within these viral reservoirs, the immune escape mutant viruses are still archived and can quickly rekindle infection as soon as ART is stopped. So that leads to us to ask the question, how can immune system effectively target HIV and especially trigger cell death if we want to clear the viral reservoirs? So we ask the question, we know that the breadth is always the issue because the virus is so diverse. And in each patient, the quasi-species of the HIV is too much for the adaptive immune system to overcome. Although both B cells, antibodies, or T cells can target conserved epitopes. And this conserved is relatively conserved. It's not absolutely conserved. So what's absolutely conserved, or I want to use the term immutable, is the HIV protein function. So we ask the question, if our immune system can recognize HIV based on the protein functions, which is immutable, then can that lead to better clearance or control of HIV infection? And we start to focus on HIV key enzymes like protease, reverse transcriptase, or integrase. And we believe that these 3 key viral enzymes, their enzyme activity is absolutely immutable. Otherwise, the virus becomes defective. So is there any immune sensors that can actually be triggered based on the enzyme activity of these proteins? We're particularly interested in the inflammasome signaling because it drives rapid cell death, which can be a potential strategy to clear residual HIV-infected cells. And briefly, our genome encodes a variety of cytosolic inflammasome sensors, and upon engagement of their ligands can trigger caspase-1 activation or inflammasome complex assembly and caspase-1 activation, which drives the maturation of the alpha beta, as well as release of the N-fragment gasmin D to form membrane pore to drive cell death. And then our hypothesis is in HIV-infected cells, when the viral proviral genomic DNA start to produce viral proteins, can such protein being sensed by a certain sensors? And if so, can the HIV target cells be trigger pyroptosis? So we started with this work by a talented postdoc, Jian Kun, and he started with a very simple system in 293 cells. So in 293 cells expressed with alpha beta and caspase-1, you can normally use these cell lines to detect inflammasome activity. So in this cell line that express pro-alpha beta and transfected with all HIV protein, you don't see any alpha beta cleavage because the cells is not provided with caspase-1, so there is no downstream signaling. And as soon as we provide caspase-1 into the culture system, the cells transfected with all HIV protein quickly trigger caspase-1 activation and alpha beta cleavage. And we know this is protease activity specific because when we add protease inhibitor into this culture, the alpha beta cleavage is fully blocked. As you can see that the HIV protease activity, which can be monitored by the HIV GAG-POL processing, is also fully blocked. So that tells us that the HIV protease is being sensed. And what's more important is it's actually the protease activity, not the protein itself, is being sensed because the protease inhibitor can fully block the sensing process. So we're really excited about this, and we went to look at the previous literature. Is there any example that inflammasome can be sensed, can sense microbial protease activity before we start to do the screening? And recently, 2 papers published to describe the mechanism of mouse NALP1B activation, which is triggered by a bacterial protease, Asrax lethal factor. And the mechanism of action is that the lethal factor protease can cleave the N-terminus of the NALP1. And if you focus this part, the fine domain part, it has the autoproliferative cleavage site. So the mouse NALP1B can actually undergo autoprocessing within the fine domain to form a non-covalent bond between the N fragment and the C fragment. So upon cleavage by the bacterial lethal factor, the neo-N-terminus is unstable and can be quickly ubiquitinated and target for proteasome degradation. And keep in mind, it's very important that the non-covalent bond is actually helping here, is that the protein undergo proteasome degradation pathway. Due to the non-covalent bond, the C fragment is actually released, not degraded, but released from the proteasome. And this C fragment containing part of the fine domain and the CARD domain is actually a bioactive domain to engage with caspase-1 and trigger inflammasome activation. In human, we also encode NALP1. And very recently, several groups reported at the same time that the human NALP1 can sense viral pathogens. For example, the human rhinoviruses, and also can sense viral double-strand RNA. However, our immune cells don't have functional NALP1 inflammasome, and also the 293 cells that we used do not express NALP1. So we move on to look at another human protein that also contains a fine domain, which is CARD8. And mouse or rats do not encode CARD8. And recent studies showing that the CARD8 can form a functional inflammasome in human immune cells, but which pathogens can activate CARD8 remain unknown before our work. So we don't know what's the function of CARD8. And we show here that CARD8 is highly expressed in all immune cells like CD4 T cells or macrophage, monocyte-derived macrophages or whole PBMCs, and also expressed at a much lower level in 293 cells. So if we knock out CARD8 from 293 cells and then transfect all HIV proteins with the presence of pro-alpha beta and caspase-1, this alpha beta cleavage can be fully blocked, very similar to protease inhibitor, suggesting that the CARD8 actually is responsible for sensing HIV protease activity. And then we further demonstrate that the CARD8 is actually the direct substrate of HIV-1 protease. In experiment here, it's a in vitro cleavage assay that we purified CARD8 protein and incubate with lysed HIV particles. And in this case, you can see this smaller C fragment of CARD8, suggesting cleavage at the 2 different sites of CARD8. And then this cleavage can be blocked if we use HIV-specific protease inhibitor here. The problem here is that in HIV-infected cells, the 3 HIV enzymes remain inactive because you can see that the virus actually produce a GAG-PO polyprotein, like here, the GP160 GAG-PO polyprotein. And within this GAG-PO polyprotein, we have protease, reverse transcriptase, and integrase as embedded form. And these enzymes do not have activity. And then all the way until viral budding, they remain inactive. And then once the virus left infected cells, then these GAG-PO start to dimerize or multipolarize, and then trigger enzyme activation and cleave the viral particle to drive viral particle maturation. So in another word, in HIV-infected cells, the CARD8 cannot sense HIV protease because it's not active. So we have to find small compound or drugs that can drive intracellular protease activation and to test if CARD8 can sense its function. About 20 years ago, Stephen Goff's group reported that actually, or very interestingly, one class of anti-HIV drugs being used in clinic for 25 years, called non-nucleoside reverse transcriptase inhibitors, can actually drive GAG-PO dimerization. And then this GAG-PO dimerization may activate HIV protease within infected cells. So our hypothesis is that if in HIV-infected cells, we add these drugs, can we trigger protease activation and CARD8 recognition and then observe killing of these infected cells? So to do this experiment, we infect monocyte-derived macrophages with HIV reporter viruses that express GFP. So 3 or 4 days after infection, you see about 10 or 20% cells infected, they become GFP positive. And then we add these drugs into this into this culture and look at the evidence of pyroptosis. And this is a drug called rilpivirine, one of the NNRTIs. Immediately after addition of the drug, you can see membrane swelling, rupture, and you see the large bubble around the GFP positive cells, which is the typical morphology of cells undergo pyroptosis and the rapid loss of GFP signal. And if treat these infected cells with other antiretroviral drugs, they don't have this function, or in the MSO group. So we're convinced that the macrophages infected by HIV undergo pyroptosis if we trigger intracellular protease activation. And then you can see these drugs can also drive alpha beta release by looking at the alpha beta concentration in the cell culture supernatant. And then this cell killing can be blocked efficiently by a caspase-1 specific inhibitors or pan-caspase inhibitors. And the uniqueness of the CARD8 inflammasome is that the cleavage drive proteasome-dependent protein degradation to release the functional C fragment. So in theory, a proteasome inhibitors can block such killing, which is also true that we tested different inhibitors, including MG132 or bortezomib, that efficiently block killing. And then we knock out CARD8 gene from the human THP1 cells, and we generate CARD8 knockout cells, and then the killing is fully blocked, which is similar to caspase-1 knockout THP1 cells. And this cell killing is not affected when ASK is knocked out, suggesting that the CARD8 inflammasome is not ASK dependent. And then the NELP3 knockout is served as a control to show the independence of the CARD8 inflammasome. And then recently we map out the precise cleavage site, which is between the 59 and 60 position. So in macrophages with CARD8 knockout, this is the inducible system by DOX. And then if we induce the CARD8 expression by DOX, we can rescue killing of these infected CD4 THP1 cells. But if the cells was replete with cleavage site mutant CARD8, like the 59 or 60 or combo mutations, induction of this mutant CARD8 cannot rescue, further confirm that the cleavage site or cleavage is important for this signaling pathway. And what's remained puzzling to us for the moment is that in these THP1 cells, the wild type THP1 cells, the cell killing is very rapid. Like you see more than 50% of the cells can be killed within 3 hours. and then reach maximum killing after at about 12 hours. And then if you knock out CARD8 or caspase-1, the cell killing can be fully blocked. But if we knock out gastamine D, the first 6 hours, we don't see any cell killing, but then afterwards, we see the killing climb up, and then actually pretty much identical to a wild type cell. So we don't know what's the alternative cell death pathway independent of gastamine D. And then initially our hypothesis was maybe gastamine E was also involved, but as you can see here, the gastamine E knockout partially delayed the killing, but the gastamine D and E double knockout cells still, died to the similar degree compared with wild type cells. So we're still trying to figure out what's the gastamine D independent cell killing downstream of caspase-1 and CARD8 inflammasome. What's more important to us or to HIV treatment or cure is to show that this CARD8 inflammasome is also functional in primary CD4 T cells. Inflammasome or pyroptosis is still a debatable or unclear feature in lymphocytes, especially in T lymphocytes. And we again use the HIV reporter system that the cells infected by HIV will become GFP positive. And when we add NNRTIs like efavirenz or rilpivirine to the culture, you can see after 1 hour, a lot of cells become caspase-1 active, as shown by a specific flick caspase-1 dye that only stain for active form of caspase-1. And this caspase-1 activation can be fully blocked with lopinavir, which is a HIV protease specific inhibitors. And this drug can fully block caspase-1 activation triggered by the NNRTIs that drive GAG polymerization. And then in these primary CD4 T cells, if we knock out CARD8, the cells are no longer died despite the protease activation, which is similar if we do caspase-1 knockout, but again, not ASC knockout dependent. As in the very beginning, we proposed that we should target immutable element of HIV, which gave us a better chance to have broader treatment or targeting strategy. As you see, worldwide, there are many HIV isolates like subtype A, B, C, D circulating in different part of the world. And in North America or Europe, the subtype B is the dominant subtype. And in Africa, C is more dominant. So our hypothesis is that if we're targeting the protease function, then no matter which subtype of HIV, they should be all susceptible to CARD8 recognition. So to do this, we started with HIV clinical isolates across the worldwide. This is the primary CD4 T cells infected by one of the HIV clinical isolate. And then if look at these control cells, if we gated on P24 positive cells, which is a dye to stain for HIV GAG antigen, when we add rilpivirine, you can see that caspase-1 is quickly activated. But if we knock out CARD8 by 2 different guiding RNA, this caspase-1 activation can be blocked completely. And then to show that this is well conserved across different subtypes, we tested subtype A, B, C, D HIV, and we do the same caspase-1 assay in control cells with wild type normal CARD8 expression. You can see that addition of the rilpivirine to drive HIV protease activation, the active form of caspase-1 can be rapidly detected within infected cells. The blue bar are cells treated by rilpivirine to drive protease activation, whereas the black bar is DMSO control. And in primary CD4 T cells with CARD8 knockout, these clinical isolates can no longer drive caspase-1 activation or cell death. And this is the cell killing, as you can see here, that efavirenz and rilpivirine can drive GAG polymerization and HIV protease activation. And both of the drugs can clear infected cells. This is the bar showing the remaining infected cells. The control drug nevirapine, which does not activate the HIV protease, in these drug-treated cells, you can see 100% of the infected cells remain, whereas when the cells treated with efavirenz or rilpivirine, especially in clade B and C, more than 70 or 80% of the cells are quickly cleared within 6 hours or 24 hours after treatment. And this phenotype is well conserved across all the clinical isolates, including subtype A, B, C, D. The last piece of the data for this story is that we want to show that this is actually a good strategy to target residual HIV reservoirs in patients under ART. So with a well-established clinical assay, we test ex vivo how about this strategy for clearing latent viral reservoirs. In HIV patient on ART, the average size of the reservoir is about 1 per million. So we call infection unit per million. The average frequency of these reservoir cells is probably 1 out of a million. And then in control regimen that doesn't have the protease activation function, you can see that the value measured in our lab is very similar to all the clinical studies. And then if we treat these patient cells with one of the NNRTIs, that result in a very quick and dramatic reduction from IUPM value 2.61 quickly reduced to 0.16, which is the average and 96.3% reduction of the reservoir. And interestingly, 3 out of 8 patients no longer have a residual HIV in their blood. Well, this is exciting, but everything is done ex vivo. So, and these drugs have been used in clinic for decades, and apparently they cannot lead to HIV cure. So that the issue here is that these drugs were developed to block HIV infection. So the potency of these drugs for blocking HIV infection is really good, with the IC50 about a very low nanomolar range. However, to drive HIV protease activation, the drug have to bind very tight to the GAG-PO polyprotein to drive the protein dimerization, which then activate HIV protease. That's why it's not effective or very effective in clinic, because the cell killing potency is much lower than the viral infection blocking potency. And the cell killing potency, the IC50 is above 1 micromolar. And the clinical concentration of this drug normally is below micromolar. That's why patients taking these drugs do not have instantaneously the reservoir reducing effect. So we have to find new drugs that can dimerize HIV GAG-PO more efficiently at the maybe low nanomolar range. Or alternatively, we can find compound that can boost this clinically approved drug in terms of cell killing efficiency. So recently, our scheme is that HIV protease can cleave the N-terminus of CARD8, release it for proteasome degradation, that then free the bioactive C fragment to drive the inflammasome activation. And then recently, Wu Hao's lab published a paper showing that DPP9 is the host factor that can sequester CARD8 through direct binding. So that can rescue the cell death pathway. And then a drug that called Vialborapro can block the DPP9 and CARD8 binding. So our hypothesis is that HIV can activate CARD8 to release the C fragment. But at the steady state, the C fragment can be sequestered by DPP9. So if we treat the cells with a combination of NNRTI plus this VBP drug, can we enhance potency and hopefully reach clinical therapeutic range? So we started with in vitro assay to show that this is efavirenz or this is rilpivirine. And the orange shaded area is the clinical concentration in patient. And then without VBP combination, it's really poor because the clinical concentration barely reach IC50. But if we treat the cells with combination of one of efavirenz, rilpivirine plus VBP, then the cell killing IC50 becomes less than micromolar. And then in this case, both efavirenz and rilpivirine, even at the 100 nanomolar or lower than 100 nanomolar, can reach 50% cell killing. So we move on to humanized mouse models to test whether this combination strategy can quickly remove HIV-infected cells in vivo. So in these mice, we infuse with HIV-infected patient blood CD4 T cells. And just after 6 hours post-cell, well, together with the drug treatment, and just 6 hours post-cell infusion, we measure residual HIV-infected CD4 T cells. And then you can see that the black dots are the control, no drug treatment. The red one is the efavirenz alone, and then the green ones are efavirenz plus VBP. And you can see that the combination treatment, efavirenz plus VBP, reached significant level in the blood. For example, after 6 hours, about average 40% of the HIV-infected cells from patient is removed in the humanized mouse model. And we also look at the lung. It's also the same, although less effective, likely because the drug concentration in tissues is lower in blood, but still it reached significant level that about 30% of the infected cells are removed by this combination treatment, as opposed to efavirenz alone, which is not statistically significant compared with the control group. So this part I want to tell you the story is that we believe this is a very conserved way to target HIV through CARD8 inflammasome. So HIV target cells, both CD4 T cells and macrophages, express high level of CARD8. And the CARD8 can undergo auto-processing within the fine domain to form the non-covalent bond between the ZU5 and UPA domain. And HIV protease can be activated by clinically proved drugs called NNRTIs. And hopefully we can find more potent drugs in the future to trigger GAG-PO dimerization, which activate HIV protease. And its cleavage at the N-terminus of CARD8 can drive proteasome degradation to release the UPA CARD domain. And the UPA CARD domain can interact with procaspase-1 to drive its activation and pyroptosis of infected cells. Since I mentioned briefly humanized mouse, I want to do some advertisement, more like a introduction of the humanized mouse model we have in the lab instead of a specific project. So our lab is very interested in developing humanized models to study HIV infection because there is no mouse model for HIV and there is no good animal model for HIV either. Monkey model can be really expensive and also slow, and the pathogenesis is quite different. And the immune escape, persistent infection are also quite different. So in reality, if we want to study HIV infection, we want to have a model that has human immune system and can test patient viral isolates. That made me interested in humanized mouse models. And what we normally start with is taking hematopoietic stem cells, which is enriched in CD34 positive population. And these cells can be acquired from fetal liver, cord blood, or adult bone marrow blood source. And then we transplant these cells into immunocompromised mice. And then hopefully in 8 or 12 weeks, we can humanize them because these stem cells can differentiate into different lineage of human immune cells. However, the human hematopoiesis is not really robust in any humanized mouse models because in conventional human mice models, normally we either have RAG1-2 knockout or skip mutations, which block the T cell, B cell development. And we also introduce common gamma chain knockout so that the murine NK cell development is also blocked. And these mice do not reject human cells, but they don't support human hematopoiesis either, because if you use this conventional model, you compare with human or mouse, you can see that in bone marrow or blood, most of the hematopoietic or leukocytes or white blood cells are myeloid origin. But in a conventional humanized mouse, you don't really see much myeloid cells. You see a lot of B cells and a little bit T cells, very few myeloid cells. That's because the other compartment is not, cell compartment is not fully developed. And if you look at the hematopoiesis, then when you put human hematopoietic stem cells into the mouse bone marrow niches, their differentiation is actually dependent on the mouse growth factors or cytokines. And whether it's cross-reactive between the human receptor and the mouse cytokine dictates how efficient the very specific lineage of hematopoietic cells can differentiate. And if you look at the, whether the human cytokine and the mouse cytokine are well conserved or not, you can pretty much predict which lineage of hematopoietic cells can be functionally or poorly developed. So in this case, if we look at the TPO, IL-3, GM-CSF, M-CSF, or GCSF, they're all poorly cross-reactive between human and mouse. That's why the human myelopoiesis process is really underdeveloped. And also the NK cells that rely on IL-15 is poorly cross-reactive between human and mouse. That's why in humanized mouse, you don't really see much functional human NK cells. So there are many ways to overcome the poor cross-reactivity to enhance human hematopoiesis. For example, you can inject cytokines, or you can do hydrodynamic injection of the DNA vector, or you can use viral vectors or transgenic expression. So these are either temporary or lifelong, but the issue is that the serum concentration often is too high for all these approaches, and these approaches do not have any tissue specificity of the cytokine expression. So while I was doing postdoc in Richard Flavell lab, we started with novel humanized mouse models in collaboration with Regeneron. We start to build these knocking humanized mouse models so that the human genome, human cytokine coding genes are knocked into the mouse genome, so that the expression of these human cytokines can be lifelong and at the physiologically relevant concentration and has the tissue specificity. So this is the scheme of the model. This is the mouse cytokine allele, and this is the human cytokine allele. And then the entire open reading frame, including introns and exons of the human, is as a whole is knocked into the mouse allele under the mouse promoter. So this is the physiological relevant expression of the cytokine. And another advantage is actually this is a knockout of the mouse alleles, which create more physical space or niche for the replacement of the human cells. And then it's actually 15 years of work now through many postdocs. We now have this model called MISTRG. I would call it MISTRG6 A215 model. So that it has the M-CSF, IL-3, GM-CSF knocking to drive the human myelopoiesis, SIRP-alpha knocking to build phagocytic tolerance, and TPO drives the long-term maintenance and self-renewal of functional hematopoietic stem cells. And this is the conventional RAG2 knockout and gamma chain knockout to block the mouse T cell, B cell, and NK cell development. On top of that, we also have IL-6 knocking, which drive or improve the hematopoietic progenitor differentiation, especially the myelopoiesis, and also improve the antigen survival of the antibody producing B cells. And then my lab is very interesting, HLA-A2 knocking, which helps the thymic selection of A2 restricted CD8 T cells, and also IL-15 knocking to drive both T cells and NK cell development and survival in the periphery and tissues. So this is the model we're using for the moment. We call MISTRG615, and compare with NSG mice, which is the standard strain in a humanized mouse field. You can see an increased level of human myeloid cells, or especially macrophages, by CD14 staining. And also NK cell development is improved, stained with NKp46. So the MISTRG or MISTRG 650 mice has much higher frequency of myeloid cells, lower frequency of B cells compared with NSG, and also increased level of NK cells. So basically they bleed like a human, which is great. What's more important is in the tissues, in a very original paper published by Anthony from Richard Lab, you can see that the MISTRG mice have these beautiful lung tissue-relevant macrophages in the lung, in the liver, in the column because of this cytokine knockin. And these cells, you can rarely see them in NSG mice. And then in the MISTRG 650 mice, we can further increase the NK cell development by knocking IL-15. So we have the beautiful NK liver rather than NK cells, which allow us to study the unique features of the liver residency of this NK cell population, and especially upon HIV infection. And the reason we focused on NK is because this is really a good model to study NK maturation, especially the tissue-specific NK maturation. This can be an issue for HIV infection because HIV mainly replicates in lymphoid organs like lymph node or spleen. The NK cells play a very robust role in controlling HIV infection in the periphery blood, but then not very efficient in lymphoid tissues because we know that NK cells in lymphoid tissues are often immature. For example, CD16 is often regarded as a maturation marker for NK cells. And then in blood, majority of NK cells are functionally mature, expressing CD16, but in tonsil or lymph node, the percent of CD16 positive NK cells are very low. And we're really happy that our humanized mouse models recapitulate this feature. In blood, lung, or liver, these periphery tissues, vast majority of NK cells are functionally mature, expressing high level of CD16. In spleen, it's an intermediate level of maturation. And in lymph node, the maturation level is really low. And in NSG, the absolute number of NK cells are really low. And because the lack of human IL-15, none of these NK cells are functionally mature. And then if we go to look at their function, by looking at the granzyme B expression upon ex vivo stimulation, you can see that the non-lymphoid tissue NK cells like lung and liver has very high level of granzyme B expression. And then in spleen, the granzyme B expression level is much lower, but then in lymph node, the lymph node rather than NK cells barely express any granzyme B. And if you provide these NK cells with the natural target K562 cells that does not express MHC class 1 molecules, then again, the same is true that the non-lymphoid tissue NK cells are more matured, have much higher killing capacity compared with lymphoid tissue NKs, including spleen and lymph node. So it's a nice model for us to study HIV infection with a tissue-specific NK maturation pattern. And then in the past, we also did CD8 T cell specific control of HIV infection. In this case, we generate patient-derived humanized mouse. We take bone marrow from HIV patient and engraft mice with this patient bone marrow CD34 positive cells, so that in 2 months, these mouse actually develop patient-specific immune system. And then we also collect blood CD4 T cells from the same patient, and then outgrow HIV from these cells. And then we can also do antigen specific stimulation of the patient CD8 T cells. So then we can use the patient autologous virus to infect these mice with the autologous immune system, and then study the patient CD8 T cell function by adoptive transfer experiment. So in this case, we specifically stimulate patient CD8 T cells with relevant or irrelevant peptide. And in this case, irrelevant peptide stimulate CD8 T cells fail to control viral replication, whereas CD8 T cells stimulate with a mix of HIV-specific peptides had very robust killing of HIV infection. And then even a single T cell clone stimulated by a single peptide can partially suppress viral replication in this humanized mouse model. So that really give us a opportunity to study a patient-specific immune response in a very unique humanized mouse model. So I don't want to get into more details about our humanized mouse work, given the time of now it's 8:55 or almost 55 minutes. So I want to acknowledge my current lab members who did amazing jobs through the pandemic, and especially the postdoc Qiankun Wang, who did the CARD8 inflammasome work. And a lot of work in my lab requires the clinical resource, especially patient bone marrow, patient blood. So our infectious disease research unit, a clinical unit led by Dr. Rachel Priesty, is super helpful to get all the patient consent, and also have our own patient cohort is really a key to our success. And our collaborators, Sabla and Carl, for help with a lot of biological and microscopy techniques. And I want to thank everybody tonight for listening, or in the morning for listening. And I'm happy to take any questions.
Hey, Liang, this is awesome. This is truly impressive, both breadth of the work and also the detailed dissection of the cellular mechanisms is great. Feel free to raise your hand or type in your question in the chat, and we can go one by one. Chuan, go ahead. You raise your hand first.
Hey, Liang. It's really great work. I think it's really fascinating. I'm not really an innate immune person, so I'm gonna ask a lot of naive questions. So first thing is, I'm curious about this HIV proteinase cleavage on the CARD8. Can you comment on the specificity on this cleavage? Whether, first of all, whether other virus proteinase can also do the similar job? And the second thing is whether HIV proteinase only specifically cleave this site?
Okay, so CARD8 is really new. We didn't even know it was the inflammatory sensor until our work. So I don't think any viruses have been discovered yet to cleave CARD8. So HIV is the only one. But I believe through more work, or when people get more people get interested in this inflammasome, then maybe there are other pathogens. And one of the directions my lab is looking at is maybe endogenous retroviruses, because it can be really an interesting idea that these herbs was actually restricted by CARD8 through evolution process, but we don't have any evidence yet. And why HIV specific cleave CARD8? I have no idea, because CARD8 evolved, million years ago. So it's apparently not because of HIV, which outbreak was 40 years ago. So the CARD8 function is definitely not developed because of HIV. But why HIV can specifically cleave CARD8, we don't actually know. But for HIV, during the natural course of infection, it might not matter much, because as I mentioned, or try to be clear, is in naturally infected cells, the viral protease is not active because it's embedded in this polyprotein. So maybe HIV doesn't encounter a CARD8 at all unless we have a small molecule to drive the intracellular protease activation. Then the CARD8 can be quickly cleaved. And the cleavage site is very specific, because if we mutate that cleavage site, it's no longer sensed. And the cleavage site actually, the amino acid sequence is really similar to the HIV self-cleavage site. So I don't know why CARD8 carry that kind of cleavage site, which is flanked by 2 phenylalanine. And phenylalanine is the highly preferred amino acid to be recognized and cleaved by HIV protease.
You have evidence that the cleavage of this HIV protease with the CARD8 is direct instead of indirect?
Yeah, because we have this biochemical assay. When we put the 2 proteins together, it cleaves. So it doesn't require anything else.
Okay. Okay, my second question is regarding your humanized animal. So first thing is your MISTRG and MISTRG 6 to 15 mice. What's the difference of these two?
Oh, MISTRG and MISTRG 615. Oh, so two more cytokines knocked in. So because I'm really interested in CD8 T cells and NK cells, so we have to have these IL-15 knock-in to further boost. The original MISTRG mice, they can also have a little bit better NK cells because macrophages also make IL-15, but only inflamed macrophage make them. So in non-inflamed tissues, the NK cell residency can be an issue. That's why we also want a non-hematopoietic cells, which is mirroring origin, also have the capacity to make human IL-15. So that's why we add more cytokines onto the original model.
So you know that I guess my next question is because you generate all these complex humanized cytokine, and then the cytokine environment or the immune milieu is not only sufficient for the hematopoietic cells development, but also very important for the organ and neuron development. I guess my question is, if you actually have this mice, when you actually start to create or let's say when these mice start to develop, do you notice any abnormality in terms of the development?
So it's always interesting is that the cross-reactivity is always a one-way street. So interestingly, the mouse can always utilize human cytokines, but not vice versa. So when we knock in these human cytokines, although there is certain degree of reduction of the cross-reactivity, but the human cytokines actually can partially restore mouse hematopoiesis. For example, when we humanized MCSF, right? We know MCSF knockout mice will have bone issues, but our mice that express human MCSF do not have obvious bone issues, suggesting that the human MCSF can actually partially rescue the mouse MCSF knockout phenotype. But when human cells are present, then the human cells are actually more competitive when they compete for human cytokines. That's why still it's a good, it's more like a knockout when the human cells are put into the place. The exception is GM-CSF actually. So all the other cytokines are partially cross-reactive, but the GM-CSF is not. The evidence is that alveolar macrophages is definitely GM-CSF dependent. So in our MISTRG mice, without human engraftment, the mouse lung only carry a little bit neutrophils, and there's absolutely no mirroring alveolar macrophages in place, which is quite interesting because you see this really funny looking lung section with, I forgot the name of the disease. So there is lack of clearance of proteins or toxins because the lack of alveolar macrophages. And once you engraft human stem cells, they actually partially rescue this phenotype. So, but beyond GM-CSF, I think all the other cytokines cross-reactive.
So you're talking about even in the, let's say, if you have just a pure wild type animal, and if you infuse with human cytokine, it will not interfere too much of mouse development. That's what you're saying?
Yeah.
And even that instead of the myeloid system or the hematopoietic system, which have compatible between the mouse and human cytokine, the other system, for example, neuron system or endocrine system, with these human, human infused cytokine in the mouse shell, it doesn't interfere the development either.
Like the, like cytokine injection or transgenic expression?
Either way.
Yeah, they don't normally cause any inflammatory response, interestingly. So it's still it's not 100% cross-reactive. So those human cytokine doesn't trigger like systemic or organ damage or anything.
No, I guess what I'm asking is not necessarily by transplant or you have infusion of these cytokines cause the inflammation. I'm talking about the system development. Let's say if these, whether these might have some abnormal of neuronal phenotype, things like that. Because there's a lot of specific organ, or let's say neuron growth, which will require the specific cytokine stimulation. I'm not sure whether when you talk about that, mouse and human cytokine, whether they still have certain compatibility, but in your system, the MISTRG system, whether you can observe any of issues.
Yeah, we never look at the brain. We never look at we never look at the brain. So we don't know. When we do infection, we do see microglia, or I shouldn't say microglia, but macrophages of human migrate into the brain, and then we see HIV infection up there. But we never look at the pathology side of the model.
Okay, thanks. Thank you.
Yeah, I think the next one is Cell A. Timin, you're the next one.
Hey, Liang, this is super exciting, and this is a milestone in the HIV research and treatment. So I'm super excited. My question, I think, is maybe more general about the latent reservoir of the viral infected cells. So is there any specific feature for the HIV, like for it to have these latent reservoir? Or is there any other viruses can maintain this reservoir under drug treatment?
Yeah, so for HIV, latency is pretty simple. If you compare with other viruses like EBV or different type of herpes virus, so for those viruses to maintain latency is much more difficult because these viruses has to maintain self-replication following the host cell division. But for HIV, it's pretty simple because it integrates into the host genome. So it doesn't have to do anything extra. And as far as latency, it's because HIV promoter, LTR, basically has all the elements to respond to immune activation. Like the LTR has 3 NF-kappa-B binding site, has 2 NFAT binding sites. So basically, it's like a T cell. It's like a gene respond to TCR. And what you can imagine is its transcription level and protein expression level can be really high in fully activated CD4 T cells, and can be quickly shut down if the cells reverse to resting memory or quiescent stage. So in reality, HIV does preferentially infect activated CD4 T cells. That's why the half-life of HIV infected CD4 T cells is about a half day. So most of the infected cells will quickly die, not necessarily due to viral cytopathic effect, but because the nature of these activated CD4 T cells. But then, as we know, the establishment of immunological memory, always a small fraction of these T cells reverse back to resting memory CD4 state, and they can become quiescent. And in this resting memory CD4 T cells, we believe that the viral promoter can also become silenced, just like most of the T cell specific genes can be silenced in the same fashion. So that's why the virus can become latent in resting memory CD4 T cells. And then because in each HIV infected individuals, there's a huge quasi-species. So they all gets archived in a reservoir that make it very difficult for our immune system to control following the viral rebound.
I see, okay. Does like monocyte or macrophage contribute to this like latent reservoir?
Yeah, definitely. So that's I think that's one of the debatable, but I think it's the correct opinion is that tissue macrophages are also very long-lived and susceptible to HIV infection. So why can they be a part of the reservoir? They definitely can. It's just without animal models, it's very difficult to study tissue macrophage reservoir by taking biopsies or blood samples from human, because monocytes are short-lived, so they're not reservoirs. You have to get to tissue macrophages, but as soon as you get tissue macrophages, they lose tissue identity, so you cannot study them. So that's why we're also interested in humanized models to look at the tissue reservoirs, especially in the myeloid compartment, but they can definitely be a part of the reservoir.
I see. Okay, that's cool. So I have a short question for the humanized mice, which is really the MISTRG, which is really interesting. So are these? I guess these are not commercialized so far, right?
Well, that's a really long story. It's between Richard Flavelle and the Regeneron and also Jackson Lab. So I think they need to work out a way to put mice at Jackson Lab so everybody can request them and get them. But also try to learn how to maintain those colonies, another issue. Because you can imagine, as Wuchuan mentioned, these mice have so many cytokines replaced, so they're definitely much more susceptible to any microbial infection. So, but the first step is make it, worldwide available to all the labs, at least in the academic setting.
Okay, cool. Yeah, thank you.
Timmy.
Yeah, hi. Yeah, sorry. I joined the lecture a little bit, the seminar a little bit late. I missed some part. So perhaps you already talked about. So actually try to ask the most of my question about the specificity, right? So I wonder whether you test whether the HIV protease can cleavage human NNRTI. Additionally, mouse does not have CARD8, but mouse has 3 NNRTI paralog. So does HIV protease cleavage any of them, right? Thank you. Okay,
so I'm more willing to tell you guys than to anybody else. HIV protease can also cleave NELP1. We haven't published this yet, and we don't want to tell anybody yet. But the issue is that, well, precisely, HIV protease also cleave the N-terminus of NELP1 at a very similar recognition site. But we have difficulty to prove that it's functional. The reason is not about the cleavage. The reason is about the NELP1 function in human immune cells. We don't know why human NELP1 is not active or does not have function in immune cells. So far, you see all these human NELP1 paper published within the last 12 months, a lot of high-profile papers. They all use the same cell line, which is the airway epithelial cell line. Quite interesting. So maybe they test through all the cell lines, and this is the only cell line that they can have the functional NELP1 inflammasome. We tried, so in CD4 T cells, they also express NELP1. We can detect it by qPCR, by Western blot. And since HIV also cleave NELP1, in theory, when we knock out CARD8, it shouldn't 100% block cell death. But in reality, it does, suggesting that cleavage of NELP1 does not result in cell death. Why is that? We don't know. And the same holds true in macrophages. They also express NELP1, but it seems like knocking out CARD8 fully blocks cell death. So how can we free, or I don't know, like lift maybe a mysterious block of NELP1 in human immune cells? Can be very interesting. We never tested a mouse NELP1 ABC. So I think B is more similar to human NELP1 and also similarly activated by microbial protease. Mouse NELP1A is also highly expressed in all the blood cells, and there was some studies showing that the mutation in mouse NELP1A can cause issues in hematopoiesis. We still don't know exactly what mechanism drive that, but we didn't test if HIV protease cleaves the mouse NELP1 alleles. for CARD8, mouse don't express those. Maybe it's due to gene duplication, or maybe the mouse lose CARD8. It could be either way. We don't know how these evolutionary events occurred.
Yeah, it's it's a great answer, right? I can tell you something. Back to 2013. So I joined the Wu lab in 2012. Yeah, I think I started work on like Trap6. Later on, the project was scooped. Then I picked the NNRTI and the CARD8. At that time, we were trying to crystallize, right? But we didn't get the crystal, right? But we had a very interesting observation at that time. So our in vitro functional assay in terms of polymerized downstream caspase-1, I found CARD8 is more potent in terms of polymerized caspase-1. But at that time, the common sense in the field is that CARD8 is not cannot form an inflammasome, it's not functional. How also think that, oh, perhaps it's an artifact, right? Later on, we dropped out the project. So after so many years, then there are particularly the 2019 paper and also a new patent, DPP9, was identified to form a complex with NRP1. So Wu lab picked up the project again and used CRIEM to determine all these structures like in a year.
Yeah, they had a nature on the NELP1 DPP9, and then had an immunity with CARD8 and DPP9. Just within the last couple of months.
Right. Initially, actually, how I want to put them together, but I think the post and students worked on this project. They want to publish it too, right?
Yeah. I think our data kind of agree that CARD8 inflammasome does not need ASK. So the CARD domain of the CARD8 has much stronger affinity with caspase CARD.
Right.
Whereas the NELP1 CARD cannot directly interact with the caspase CARD. So that requires the ASK. So human NELP1 is ASK dependent. But interestingly, the mouse NELP1B is not ASK dependent. So this is really confusing.
Right. Yeah.
So which one give rise to which is still, we're still trying to figure out what's going on here. But at least it's not because of HIV. That's what I know, because HIV is too young for this.
Yeah. Thank you. Yeah.
Sure.
Great. I see some old friends' name in the participants. If you have questions, please feel free to unmute. I can ask a couple more questions. One is that in the beginning, it's really interesting. You showed in 23 T cells, you don't need to add this drug to trigger a dimerization of the protease, but you can still induce caspase activation. But in monocytes, you do. Do you think
in CD4 T cells we still need?
You still need to.
Yeah, we still need. Yeah, but
both, but not in 293 T cells.
Yeah, in 293 T cells, because that's transfection.
I see. So expression dependent.
GAG-POL drives spontaneous dimerization.
And
go ahead.
In natural infection scenarios, what's the condition that you're activating this HIV proteinase?
So we just have to add the non-nucleoside reverse transcriptase inhibitors. So this is a class of clinically approved anti-HIV drugs. So the drug was by design to bind the RT, the reverse transcriptase part. And upon binding, the drug blocks the RT activity. And then as a side job, nobody initially cared, is actually the binding to the RT region of the GAG-POL actually drive the GAG-POL dimerization. So the dimerization actually mimic. So now people get interested, and then we have a collaborator showing that the GAG-POL dimerization naturally occurs within a budded viral particle. So they can get crystal structure of GAG-POL dimer within budded viral particle. And then the crystal structure is pretty much similar to the GAG-POL dimer driven intracellularly by NNRTIs. So NNRTI is more like mimic what's happening after viral budding to drive protease activation.
It's interesting. Do you think this may be a way HIV evolved to reduce detection mechanisms of their infections?
Yeah, because we don't know these early strains. So, there's a lot of history about HIV research. So the first outbreak is 1981. We do have isolates from that age, from 40 years ago. But from the evolution study, people more believe that the real HIV pandemic starting 1921-ish in South Africa, because based on the molecular clock. So, but then those sequences will never be, recovered. So around 1920s, 1930s, 1940s, was there any activity, drive the CARD8 escaping mechanism? We don't know. It's possible that the virus has to keep the protease inactive.
That's really, really interesting. And going back to the question with the CARD8 knockout conditions, do you also see IL-1 beta secretion or secretion in CARD8 knockout cells after HIV infections?
We can see it in infected macrophages, but CD4 T cells don't make IL-1 beta, so we don't see them.
Right. But in macrophage, you do see IL-1 beta secretion in the CARD8 knockout cells.
Yes.
And the cells stay alive.
Yeah. Well, they also undergo pyroptosis. So what's interesting is if we knock out gastamine D, the cells do not die within the first 6 hours, but we still see a lot of IL-1 beta release.
Right. I see. That's interesting. Okay. There's a lot of like interest now, like looking at regulation of just different pore formation or different way that you can allow IL-1 family members to be secreted without causing cell deaths.
Yes.
I'm just curious if you're happen to see this is the alternative way that you're causing IL-1 family release without triggering cell deaths in this particular scenario.
In this particular scenario, I think both happens simultaneously. But if you, like I said, if you knock out gastamine D, the IL-1 beta is still released. So that might be a good model to study this segregated event.
Right, that's really interesting. And the MISTRG 6 to A15 mice, your mice. I'm curious. So normally L6 is not majorly expressed at baseline, but in your case, you bring L6 seem to be really promoting some of the homeostatic hematopoietic differentiation process.
Yeah.
Which cells do you think highly relies on L6, and why that's the case? Where do they see L6 being made?
I think the myeloid progenitor cells can be differentiated through IL-6. I also think that when we replace the mouse IL-6 with the human one, the mouse myelopoiesis is blocked to a larger extent. So I think it's like both mechanisms contribute to improved human hematopoiesis. And then I think Richard has another paper looking at IL-6 and antibody production, but we're not studying antibody and B cell response, so we don't look at those part at all. But we're just trying to use IL-6 to further enhance hematopoiesis.
That's really interesting.
I think the murine bone marrow stromal cells make IL-6 at steady state. And then human macrophages obviously can make a lot upon HIV infection.
Yeah. That's really interesting. Thanks, Liang. Any more questions? All right, if there's no more questions, thank you, Liang, so much for this wonderful talk. It's really exciting Saturday evening here in the US. I'm sure very nice Sunday morning there to kickstart with a great weekend day in science. So hope everyone have a great weekend for the rest of the day. See you guys next Wednesday.
Thanks. Bye.
Bye.
Yeah.